If anybody who wants to stay can stay. We'll have a short break and then we'll do the meeting afterwards. So we have a lot of new people here. Everybody raise your hand who's new. You guys? Yeah, great. Thanks you guys for coming. We appreciate it. Nice having you. Our program tonight's going to be really good. We've been looking forward to it. We had Dr. Zach Boles here. He's an assistant teaching professor at Rowan University and assistant curator of the Edelman Fossil Museum Park. That's over by my cousin's house over there. It lives in his backyard. He has a BA in geology from North Carolina State and a PhD in biology from Drexel University. So we'd like to welcome him, and we're looking forward to the show. And if anybody gets a chance, I've said this to a lot of people, that museum is well worth your time to go and see. It's spectacular. Right.
So thank you again for having me and for everyone showing up tonight. So I'm going to talk about some of the research that I've been doing and some of our undergrads and new grad students have been doing at Rowan and Fossil Park. So have any of you guys been over to the Fossil Park? Cool. Yeah, so we just opened up less than a year ago, but the site itself has been there since about 1925. An Emerson company purchased it, and then West Jersey Marl actually owned parts of the property going back to about the 1880s. And then Rowan stepped in and purchased the property around 2016, and then got a very generous donation from the Gina Edelman, Rowan alumni. So we have this like gorgeous $75 million museum. So today's talk, talking about one of our recent papers from 2024, and then lots of turtle stuff. So pathologic sea turtle, we have turtle histology, turtle pathology, bird stuff, and then some other things that we hope to get done this semester. So this is the quarry itself. This is a very old photo now. This is back when we were all grad students, so this is probably like 2012, 2013, 2014, somewhere in that range. Dr. Ken Lacavera in the center there in orange, and two colleagues, Paul and Kristen, who teach at up in North Dakota. So starting in 2012, the Emerson company cleared off a big area for us to excavate the main fossil layer. So that's a bone bed that spans kind of the impact event. So our main hypothesis is that it is a mass epicenter related to the asteroid impact. So basically like an ecosystem that's slowly collapsing, all these animals dying off as food becomes more scarce and their environment changes. So starting in 2012, we cleared off this big area, and then since then we have been digging systematically through over 250, 300 square meters, somewhere in that range now. So we mark off square meters with just rebar posts, that's what all those orange bits are, and just slowly dig through the fossil layer, collect everything we come across, and map all of the bones and teeth. So today we've had over like 100,000 bones or fossils from this layer. So from all of this collecting we've been doing, a lot of really tiny teeth and little things that we've discovered. So in 2024, we finally got around to publishing on a lot of these newer, unique, important specimens. So this paper probably took about a decade to write. So we find something cool, like a small weird tooth, identify it, set it aside, and then after a while we had enough specimens, so we just sat down, wrote the paper, and got it published. So no like brand new species to science, but we did find a lot of new specimens unique to the fossil part, and we a few kind of range extensions, either stratigraphically or geographically as well. So these are a lot of the cartilaginous fish, mainly sharks, and then a ratfish can be there. So see things like shark Halogaleus, Subtrancheus, Notodenodon, Loconia, and Pseudothorax are all new species to the fossil park itself. And then a few other ones where we just kind of either modify their stratigraphic range or geographic range as well. Speaking of this lumberni here, this one at the very bottom. In North America, there's only 3 known specimens, 3 reported ones, at least all from the fossil park. New Jersey State Museum found the first one a couple years ago, published on it. We found the second one in our research grid in a fossil layer. In Jason, when we found this book, so it's sitting on the surface. Ours has stratigraphic data. And then the third one here that's figured was found by a 5-year-old girl at one of our dig days pre-museum. So that one was like really excited. So she like walks up to us and like, I know right away it's like a super rare tooth. So for that one we had to keep it, but we 3D scanned it, gave her a 3D printed one, and then her name's associated with the specimen at the museum, and then she's mentioned in the paper as well. Yeah, I think it's like one of maybe 2 fossils we ever had to take from a visitor. So lots of cool sharks, and then as you can see, like some of these are super tiny. Like top of the Halogaleus, the teeth are like 1/2 centimeter at most. Seeing this, it's close to like 1.5 maybe 2 inches. Yeah, a lot of them are very tiny. And these are some of the fish we found. So mostly little tiny scales, some they're seeded. We have Crabula as well. So fish with these kind of circular, spherical crushing teeth. So next we have Sarcocephalus, letter E right here. It's the only tooth we have. It's kind of this weird large fish. It has, at least some species, like a spike on its lower jaw. F and G are Encodus spadiolus. So Encodus, saber-tooth salmon, saber-tooth herring. Super, super common. So if you ever go like fossil collecting in these streams around South Jersey, you'll probably find a bunch of these. And there's at least like 22 known species of Encodus. We have Encodus ferox and now Encodus spadiolus at the fossil park. The teeth look very similar. And the main difference between those 2 species is that Encodus gladiolus has a little tiny bar towards the tip. H. rhabdosius, H is a gar, and then Yotis. Bunch of new fish to the fossil park. And then this is some reptile remains, croc and mosasaur. So A is the tooth of a Bobosaurus, so alligator, crocodilian. Very large, robust jaws and really robust conical teeth. So these things are eating probably turtles, clams, hard-shelled prey, using those blunt teeth to crush them. So we have that tooth, and then we have B up there is a very, very tiny tooth, and then C is the lower jaw dentary of a very small juvenile Bobosaurus. So the jawbone there on the front half of the lower jaw is maybe slightly larger than the Bobosaurus tooth in A. So yeah, it's a really small, very young individual, definitely the smallest known specimen. I think all the other ones, at least that I'm more familiar with from New Jersey, are at the Academy of Natural Sciences and NJSM, and those are all like really big, robust specimens. So that's a really cool animal. And then, yes.
Yeah, quick question.
Yeah.
The Cretaceous Paleogene layer or
Yeah.
border, is it well defined? Like below this is all Cretaceous, above this is Paleogene?
So yeah, in the Hornerstown, basically the fossil layer itself represents that transition, but yeah, there's no clear like marker thing like point at since like this is Cretaceous to Scandian. So we just splits the boundary right at the base of the fossil layer.
Okay, thank you.
Yeah, if you go below that, like in Navasing, that's definitely Cretaceous, and then like Vincent Town and like Upper Mars, definitely Paleogene. Yeah.
Is there any of the ash layer presence?
No, we have found shocked quartz, spherules, and there is a very shallow, small irradiance spike through the fossil layer itself. So we have impact indicators from the asteroid impact and the fallout. We don't have like an ash layer or anything like that because it's all marine.
Mm-hmm. That makes sense.
Just two quick questions. First, I think you mentioned this, but you said this ecosystem was slowly collapsing, not rapidly collapsing.
It's hard to say
Oh,
like
okay.
how long
Maybe
the fossil layer it
Okay.
took. So I mean, like some of these animals, there's probably enough food for them to survive. Whether that's like weeks or years, decade, weeks, months, years, we don't really know.
Okay.
So
Like
it's
not,
probably-
but that's still pretty short. Like.
Geologically speaking, yeah, it's a
Okay.
really,
Okay.
still pretty short period of time. So there's probably some animals that are already dying off, already dead, and then this ecosystem collapsed and then all
Okay.
animals
But not
die.
like a, they're dead.
Yeah, I'd say it's more kind of like more of a drought where things are slowly dying off over a period versus like a volcanic eruption where things
Right.
like very.
Right. And then my second question is, by smallest known specimen, do you mean like tiniest rock?
Just
Like
like
that-
smallest known biosaurus.
Okay, so like the creature itself was
Yeah.
small, not just like the specimen itself.
Yeah, this
Oh,
is
okay.
like a little baby
Okay, got
biosaurus.
it.
So yeah, all the ones in them are probably like adults. You know,
Okay.
really big.
My bad.
Yeah.
Okay, thank you.
And then the final one here is the pterygoids of Mosasaurus ackmani. So Mosasaurus ackmani is one of the largest known mosasaurs. Size range was maybe 50, 60 feet, depending on like what formula you use to estimate size, but definitely really big. And then yeah, pterygoid is the extra set of jaws they have on the roof of their mouth. So that one's nice, big, more or less complete. A little beat up, so this, when they're clearing off, and percentage clearing off our dig area, I think they missed that by like maybe an inch. Because when we eventually found it, it was like right at the surface. But, yeah, that one itself is probably like 9 inches across. Yeah, so we have that one, a few mosasaur vertebrae, and then a few teeth as well that have been collected from our dig area. But if we could dig deeper, we'd find probably some bigger, better, more complete specimens. So, pretty complete, very large, maybe like 5 or 6 foot skull. The mosasaur came out of the old quarry way back in, I want to say like the 50s, and that was at the New Jersey State Museum. That came right out of the Navasing. So beyond just finding specimens that are new to the fossil park, because we had like stratigraphic data, so like what layer these fossils came out specifically, we could also show that a few of them were either present in the latest Cretaceous and survived the extinction event, or vice versa. They were present in Paleogene and then in the Cretaceous. So in this chart here, you have Navasing down here, Horner Sound, and the MFL. So anything above the MFL is Danian or Paleogene in age. Anything below it is Cretaceous. So we can see that Ichthyotis and Pseudothorax, so ratfish and one of the sharks, survive a little bit into the Danian, so they survive the extinction and go extinct a little bit afterwards. Same with Encotis and Anamiotis. And then we have new Cretaceous records for Paleogaleus and Brabia. So they were already present in the Cretaceous and then survived as well, and then make it a good fit into the Paleogene.
Sorry, I'm not a fossil person, I'm a mammal genetic person.
Okay.
What are they?
So these are the sharks and fishes.
No, no teeth, what?
Oh, no, the Nematodon.
What is it?
Yeah, that's one of the sharks.
A shark?
Yeah.
Okay, what's the Cercocephalus.
Fish, and then Pylos is another fish.
And what is the, and what's the last one?
A fish.
Thank you.
So yeah, stratigraphic range distribution showing that some of these species do survive the extinction events, at least for some amount of time. And then we also have the first record of the shark Edelmen from the Western Hemisphere, Sarcocephalus and Pylos from kind of the east coast, northeast of the U.S. Yeah.
Sorry, this
Okay,
is these are my questions. So I just don't know if you know this, but are the specimens larger the deeper you go down because they would have died sooner, and then the smaller ones are more towards the top layer because they would have survived longer, or does that not really happen? So you mentioned that deeper down you would find better, larger specimens, you think? So I didn't know if that was from the dying like speed, or if that was
not necessarily in that case. Other researchers have looked at so you can see these are like little lobster burrows. So the sediment is really heavily bioturbated. So there's lots of little lobsters that were living on the bottom of the ocean, creating all these burrows. So what's cool is that there's a researcher at Temple years ago did a master's project. He measured the diameter of all those burrows going from Upper Hornerstown down to the MFL, down into the Navasink as well. And what he found is that the diameter of the burrows, once you hit the fossil layer and above it, get a bit smaller.
okay?
So for some reason, right at the boundary, the shrimp or lobsters are getting a little bit smaller. And then we see above our fossil layer, different, more primitive species. So some brachiopods and a few sponges and things. So it goes from really, really diverse, abundant species in the fossil layer and then hardly anything above it.
You don't see that type of size difference with mosasaurs. You don't see bigger mosasaurs deeper in time, and then they're tiny at the boundary.
No,
Okay.
the extinction is pretty sudden, so they get, I mean,
They're
there's,
tiny.
no, I don't think there's any trends for them,
Okay.
but, I mean, there's a good size variation for different species, so you have ones that are maybe 10 feet long, and then mosasaurs is probably 50 feet tall, so.
Okay, thank you.
What is happening between the Navasink and the Hornerstown? Geographically, what is it, is change in sea level or change in sediment type or?
Yes, so it goes from kind of more of a chocolatey marl down here to kind of more pure glauconite. So if I'm remembering correctly, it's sea levels getting a little bit deeper as you go across, but there's not much change though.
Okay.
It's still at the bottom of the ocean. There's a couple sea level changes, rise and falls throughout this time. So that was that paper. And some other cool stuff we have. So sea turtles tend to be the more common, big thing we find, followed by crocodiles. This was a really cool specimen that was found way back in 2018. It's on one of the last days we were out in the field, getting ready for the winter. So our collection manager, Mike Heyerbacher, was doing some prospecting back in one of the little gullies, found a few bones eroding out of one of the upper Hornerstown. So these are fossils and more 60-ish million years old. So I kind of dug around, found more parts of the turtle, figured we couldn't really let it sit over the winter because it was kind of being undercut. So if we had left it, it may have been washed out and destroyed. So this is from December. So we're out there super cold, putting plaster and burlap on this turtle. But it's really cool. Once we crafted it, we started to see some weird things on it. So folds, depressions, bony growth, things that shouldn't be there. So figure in the top right there, the white are bones we have and identified. The red areas are the pathologic bones, which are shown in the next couple of slides. So decently complete for specimens. So hopefully it shows up a little bit, but the arrows kind of cover up some of it. So pathologic fossils, that's one of the ribs that makes up the top part of the shell of a turtle. And yeah, it's coming up, but right there, kind of the bottom lower left of that blue arrow, there's some pits there, and then on the bottom side, there's kind of like bony growth. Peripheral there in the middle, that's like the outer edge of the shell, and then right in the middle of that arrow is just this kind of V-shaped depression. And then on this other costal, there's a depression on the outside of the shell, and then on the inside there's like this nice bulge. So it looks like you just like took your finger and pushed straight down through the shell, and then just like you like Play-Doh. Yeah, so we're not sure, and then this is the cooler piece. So this big hole right there shouldn't be there, and then at the top you can see this half moon shape. That's another hole, and there's lots of depressions all over the surface of these two costals as well.
Do they resemble tooth punctures?
So we're not sure. The one on the peripheral has kind of like a pointed bottom, so it could possibly be a bite mark. Because we have like so few of the costals making up the shell, it's really hard to see like, is there kind of a clear like bite mark pattern on them? If they are bite marks, it'd be more like a Boles-saurus or a mosasaur maybe. Definitely would it be the sharks.
Is there evidence of healing on any of them?
Yes.
So you have an injury and some healing
Yeah.
of some sort.
Yeah, so on the top of the shell, the outer surface of the shell, you have those like pits and depressions, and on inside you see like kind of where bone's being deposited directly
Okay, so
beneath
there's a
it.
deposition on the inside.
Yes, and then on the holes, on the inside of those holes, that's all smooth, fresh bone. So it's not just like some damage that occurred after death. So yeah,
So
something
whatever happened,
penetrated it.
so whatever happened, that turtle lived after that
Yes.
incident.
Yeah. So yeah, this animal survived for some period of time, enough time for it to kind of start healing and like trying to deal with whatever was going on. So turtles today suffer from lots of different infections and shell rot. So here's a photo I found earlier today. So some shell rot on diamondback terrapin. We can see that all these open sores on the outside of the shell of this animal. So this could be caused by just any type of damage. So it could have been bitten by something, some sort of damage to the shell, bacteria, fungi, something gets up underneath of it and just leads to an infection. And then if it's really severe, it starts getting to the bone itself. So with our particular specimen, we don't really know what caused it. But it seems to be kind of some sort of infection, like shell rot, if you will. But hopefully we'll be working with some like modern exotic vets sometime soon to help kind of like nail down what type of infection this is, and if we can like assign like a specific disease to it. But yeah, we have a new vet school at Rowan. They do some exotic vets, or exotic animals. Some more turtle stuff. So turtle bone histology. So this is basically taking a bone, in this case turtle shell pieces, cutting them up, embedding them in like resin, making them really thin, ground down so they're basically almost like transparent. So you can examine like what the bone structure looks like. So with our fossil park, we have about 6 known species of turtle. In the fossil layer itself, they have a wide range of sizes. So the smaller species are maybe like 9 to 12 inches for the shell. And then the bigger ones we've collected have a shell diameter of about 2 feet, and some be a little bit size range, and then the shell morphologies vary a lot as well. So some are kind of like really high domed shells, others some more flat with lots of spaces, empty space in between some of the elements, which all suggests they're probably living in different types of habitats, but we find them all together. So this was actually part of my PhD research, looking at some of the habitat preferences of these different turtles. Luckily, Torstenshire, another researcher, had already shown that if you look at the microstructure of turtle shells, it actually kind of relates to where they prefer to live. So I took a bunch of specimens, mainly stuff we had, something from the Academy, as well as the Yale Peabody Museum. So I got some just little turtle fragments, cut them up, made them super thin, mounted them on glass slides, and ended up with these nice thin sections. So you're looking at just like a cross section through one of the shell bones here. So the one on the left is Taprosis. That's one of the really common species we found at the fossil park. We have several of them now, some really small ones all the way up to like pretty large ones. Atticus in the middle, and then this is Euclastes weileni, another super common turtle we have at the fossil park. So in the figure you can see this as like Caterpillar panda, but it's actually Euclastes. So that one was renamed by my colleague, Edelman, a couple years ago. But this particular species has gone through like 20 different names, because they were all named by like, Cope and Marsh back in like 1800s. So researchers would find like a turtle bone, name it one species, someone else would find like a different part, and then that gets named something else. So as you go through and find more complete specimens, they get renamed again and again. So when I started, I think this was known as Dolicheles, then it got renamed Caterpillar, and now it's Euclastes. And then these are the specimens, thin sectioned and then converted to black and white. So black is bone, white is empty space. So what Shai was able to do is kind of categorize all the specimens he looked at, all the modern turtles, and categorize them in one of four things. So category 1, terrestrial turtles. So ones walking around on land. They have like really thick, dense shell bones, so kind of similar to like CB and E. Category 2, semi-aquatic, so spending a lot of time in the water but coming back onto lands as well. Category 3, fully aquatic, so that'd be most of your things like sea turtles. And then category 4 is extreme adaptations to open marine ecosystems. So from my research, we found that Atticus and Agathis are probably category 2, so semi-aquatic. So they're probably living more like onshore freshwater environments, lakes, rivers, things like that, which was what we expected. And then the rest were fully aquatic for extreme adaptation. So Caprosis and this close relative Bothrymnes are there, and then Paratresius is kind of weird turtle with kind of like spikes along the midline. Getting close to that category 4, and then Euclastes is category 4. So, which kind of leads us kind of wonder. So what we find most common are Taprosis and Euclastes. We have several individuals of both those species. So Taprosis category 3, fully aquatic, so probably living more along the coastline. Euclastes is more adapted to living out in the open ocean. But we're finding large numbers of both those together. So that's kind of an interesting question as to why that is. The other ones, Atticus, Agathis, and Paratresius, really rare. We have one Paratracheus specimen in our collection, no Atticus or Gompus, those are at the Academy. And then we may have like 1 or 2 pieces of Bothrones.
Real quick, while you're on
Yeah.
that other screen, which
Mhm.
ones are an example of category 3 or 4? You pointed out
Yeah.
that C, D, and E were land.
Yes, so category 4 would be like J and K there. So there's no like kind of thick outer cortex.
Okay.
So category 3 would be like these two down here. We have kind of a thick outer cortex of bone and then kind of a thinner internal bit.
Okay.
But category 4 is like really, really porous bone.
Okay.
So like hardly any like outer thick cortex. And then F is the Paratracheus, the kind of wavy outer surface. So again, kind of thicker outer layer and then a really thin inner layer.
Okay, thank you.
Yeah. Unfortunately, that one didn't load, but that one, this slide was going to be a poster that one of our undergrads did a few years ago, but we have a very, very tiny taphosis, which is maybe that big, so 5 or 6 inches per shell length. And then the ones we have on display are about 2 feet, and we have a few individuals that are in between. So we have like a pretty good natural growth series for taphosis and then maybe classes as well. Going back to the histology and habitat preference, we wanted to look at, because we have this nice growth series, are they kind of changing habitat as they get older? So today, sea turtles, like when they hatch, they like swim out like the middle of the ocean, grow up there, and then kind of return to kind of more coastal environments. So we wanted to see if maybe it's these prehistoric sea turtles are doing something similar. At least initial data does suggest that. So the juveniles have actually come with a little bit thicker, denser bone. And then as they got larger, they became a little bit more porous. So we're not, at least initially that would suggest that they might be kind of changing habitats as they're going from like small individuals to larger ones.
Zach,
Yes.
is that a buoyancy adaptation? If you're thinking about a small
Yeah.
turtle, okay, you could expect a thicker bone because he's also got lungs and that
Yeah.
sort of thing. And that's going to add to his buoyancy, right? As he gets bigger, then the volume to surface area changes by 1 power, right?
Yeah.
So you would expect that as he gets bigger, if he's got to maintain the same buoyancy, that he's going to have to get rid of some of the thick shell.
Yeah. So, good point. Yeah, so I don't think anyone has looked into if shell histology changes from
Somebody
juvenile to...
could actually calculate that.
Yeah.
Somebody could actually calculate that on their displacement and see what that looks like.
Yeah, because I think in some other, I think it's plesiosaurs, you do see that like juveniles have like pretty dense bone, if I'm remembering
Right.
correctly. Dense bone, and then as they get larger, they get more porous.
Yes.
So yeah, it could be density thing as well. Yeah, this is another research project.
Sounds like it.
Yeah.
Quick question.
Yeah.
You know how like human, we have bones and bone marrows are making our blood cells and everything. So what are the functions for the bones in turtles? I know like the shell is actually the true backbone for the turtles. So like, what are the functions for the turtles? Like these spaces that we see.
Yeah,
are
so
there,
you
like, soft
sometimes get like
tissues in there?
narrow blood vessels and soft tissue and stuff in those areas, so the same as our bones.
I see.
Yeah, it's the main function of the turtle shell is most likely protection, some other adaptations. But yeah, all the empty spaces in life have been filled in with blood vessels and tissue and marrow and stuff like that. Yeah, I want to see that poster, I can always show you guys afterwards. And then turtle taphonomy. So taphonomy is the study of basically everything that happens to an animal from the time of death through like decay, fossilization, so when it gets fossilized and discovered. So getting back to our turtles and other specimens, other species, we see kind of a wide variety of preservation. So we have like nice articulated specimens here, so that's articulated Saprospis. That's the one that's on display at the museum if you've been, this the bowl-shaped one. There's articulated specimens associated with disarticulated ones. So top here, the kind of lighter bits, those are all bits of sea turtle shell from Clastes. So we got like a clump right there, there's a few pieces in the back there, and then another clump of bones on the right. So that shell's kind of been disturbed and spread out over a few meters. And then we find like isolated bones, so that's the lower jaw of a sea turtle, and then that's like some bit of shell bone that's really poorly preserved. What's cool is that at least a few cases we've been able to find like two bones that are separated by several meters, and they fit back together perfectly. So we're trying to figure out like if something's like isolated or partial skeletons. There's more to it in our collections. We just have to go through our maps and see like which ones might be belong to the same species or same specimen. Unfortunately, because of how mixed up some of the specimens are in the fossil layer, it's a bit difficult to do. So they have found in the past like a turtle shell and a turtle skull right next to it, and they're completely different species. So things are a bit jumbled, so it makes understanding the bone beds a little bit harder. So we have this kind of wide variety of preservation of our turtles and other species. We're mainly interested in the turtles. So a current project, hopefully we get started this semester. Mainly Joe Deckut in the back here. He's our new paleo PhD student at Rowan. So he's going to be working on this project, but we want to know basically how do sea turtles decay in aquatic settings. So, if they die floating in the ocean, do they decay at a certain rate or in a certain sequence? So these studies have been done with lots of like kind of terrestrial animals and a few marine ones. They looked at like crocodilians, alligators, a few lizards, a few other things. For sea turtles, a handful of studies, mainly kind of more onshore, so like in the tidal zone, and a few other studies, but nothing super conclusive. So these are some questions we hope to answer. So how long does it take for turtles to kind of disarticulate, fall apart? Are there any trends in disarticulation? So do like certain body parts break down, decay, fall apart faster than others? So like for some organisms, like the lower jaw is one of the first things that will fall off. Does the turtle shell affect any of this process? And then like does burial exposure also affect like how they're preserved in the long run? Yeah.
You set up a tank and you put a turtle in there and you can scientifically measure the rate of decomposition.
Yes.
I know that there was this thing, and I don't remember the name of it, but they would take people that donate their bodies to science,
Yeah.
and they would study how long that they.
Yeah.
Yeah, it's Tennessee. That's right.
Yeah. So same thing. Yeah, we're just looking at.
I have a question. Is the shell the first thing to just start to deteriorate?
We're not sure. That's one of the things we want to look at. And because like the turtles, their shells are variable, so sometimes like underneath all the like keratin stuff, they're like open spaces. So kind of see like if how like well built the shell is has any impacts on how they decay, but
What about the salinity of the water?
I don't think it have much impact, but
Follow up for this, after you finish this idea.
yeah.
So my question is about the ecosystem decay. I don't know anything about decaying sea turtles. It's just a random thing, but so do creatures not eat them? Like, would they not be eaten by creatures quickly? Would a situation where the ecosystem's collapsing and they don't have enough food alter the decay process of the turtles? Or is it, for the most part, when they're floating out there, it's little like tiny guys that are eating them, and big guys are like, that's gross?
So I imagine like sharks and things are probably feeding on these carcasses. So we see it in like modern environments, so, like things, whales, anything dead living in the ocean, sharks.
But most of the like
birds, but insects, but
specimens you're finding in the fossil park, those are not like turtles that have been eaten and then their shells are just getting thrown around and their skulls are getting thrown around by different guys. They're like decomposing and kind of in the same.
yeah, that's kind of what we're trying to figure out with this project.
Oh, okay.
So we mainly find like just the shells. We have a few lower jaws, one partial skull, but it's mostly just like top and bottom of the shell.
Okay.
A few like upper arm and leg bones, like femora and femora, but like distal, like lower elements, we don't find any of that.
Okay.
Neck vertebrae are very super rare, so it's mainly just the shells
Okay.
that we are finding. So yeah, they're probably like, these turtles are like floating on the surface. Sharks, most source of the things, probably eating the head and flippers as they're kind of like hanging down in the water column. But yeah, for this particular study, we're doing kind of like ideal, like nothing's really scavenging it beyond like bacteria and insects.
Okay.
So it gives us kind of a like best case scenario. Yeah, otherwise, yeah, we would be jumping on it.
And this is it. I don't know anything about it, so I'm kind of fascinated by the question. When it comes to marine ecosystems of the Cretaceous and Paleocene, am I old?
Yeah.
Are those marine ecosystems significantly different in terms of their microorganisms? Like how for sometimes in ancient history periods, certain things decayed? I don't know, like, or is it not that different because it hasn't been that long? If you don't know the answer, you can say, we literally don't know.
No,
I just don't know enough to know.
It might be like different species, but probably doing the same thing. So
Okay, so
there would be like starfish, sea urchins.
Okay, so you don't have to
Lobsters.
like build a new like
No.
ocean that replicates the old ocean.
Yeah,
you
we
can use
just-
this ocean.
Yeah, we just want to see like in the water how do these things
Okay,
slowly
okay.
decay. But yeah, we do have evidence on some of our fossils. We have some shark tooth bite marks on one turtle shell, and then evidence of like sea urchins, kind of like
Okay.
probably eating either soft tissue or like sponges growing on the bones.
Okay.
Quick question here.
Yes.
When you describe like same procedure as the other paper, like,
Yeah.
what is the procedure that you're talking
Yes, so next slide here.
Okay.
Simon Salisbury back in 2014 did an experiment with crocodiles. So they had a bunch of juvenile crocodiles that can be like 2 or 3 feet in length. So they used one of the limbs for a different study. So they had these carcasses laying around. They put them in aquaria and had kind of 3 different methods they were looking at. So for 2 specimens, they just buried it under sediments and then filled the tank up with water. Second scenario, they let the alligators kind of decay, float to the surface, and then once it sank back down, they buried it. And in the third category, they just left them to like decay, never get buried. So they wanted to look at like, how do these crocodiles decay just in an aquatic setting, and then if burial has any impact on how well preserved the specimens are. articulated specimen of something, it usually suggested that they got buried really quickly right after death. So there's no chance of like scavengers coming along and like tearing off an arm or leg. So yeah, they just let these crocodiles decay, and then some figures they got. So after about 4-ish days, there's enough decay gases in the carcass to kind of allow them to kind of float up to the surface of the tank. And then they were there for, as you can see, like a few more weeks before either like those gas bubbles kind of ruptured, and then all the gases escaped in less point, so they sank back down to the bottom of the tanks. And then took about 2 months for kind of the carcasses to entirely decay. So the only like scavengers that were allowed into this were just like flying insects that could like get in there and lay eggs. But what they found is that the carcasses stayed together pretty well. We saw some disarticulation with mainly the pelvis area, it's like the hip region, just due to like the insects and like maggots and stuff getting the tissue. And then once they sank down and hit the bottom of the tank, which is only like a few inches down, they actually saw like a good amount of disarticulation occurring just from that like gentle impact. So these are some diagrams from the paper. On the left was one of those crocodiles they buried right away. So these couldn't like come up to the surface, decay, sink back down. And then these other two pretty similar. So they were allowed to float to the surface, decay, and then sink back down. And then yeah, just by hitting that on the bottom of the tank, bones got spread around. So pretty useful like information when you go back to look at the fossil record. So again, it kind of confirms that like really well-preserved crocodiles most likely got buried pretty quickly, but disarticulated ones, usually you would think that those things set out for a long period of time. Scavengers came around, knew stuff about, they're staying on surface for a long period of time, getting spread apart. But at least for like aquatic settings, it could just be like settling on the seafloor that causes a lot of disarticulation. So kind of the timing or how quickly these animals buried can kind of vary. So basically we want to do that same exact study but with turtles. So our idea at the moment is to just salvage diamondback terrapins, because they unfortunately can get caught in like crab pots and other things down the coast. So ideally we can kind of salvage them soon after they died and then use them for our experiments. But basically the same thing, get some aquaria, fill it with water, bury some of them right away, let other ones kind of decay, let those gases build up, let it float on the surface, and then see what happens. And then if we can, I'm trying to get like some sea turtle specimens. It's been a bit more difficult. So those would be like salvaging ones that, you know, like hit by like some boat injury or just kind of perish for one reason or another. They get kind of rescued all the time all over the East Coast. But yeah, if we could get some sea turtles, that would be awesome. But yeah, we'll inject diamondback terrapins because they are, carcasses are pretty easy to come across. And then also they're more aquatic turtle species, and at least superficially their shells kind of resemble some of the species we have at the fossil park. But again, that'll be Joe's, one of his research projects for his PhD. So hopefully later this year we'll have some results for you guys. And a few more things for finish up. So this is another really cool specimen that we found years ago. Got super lucky. The student who found it was a Drexel undergrad who's now, I think he's done with his residency, so he's like a doctor, surgeon. But he found this tarsometatarssus, so foot ankle bone of a bird in our bone bed. So this was found years ago. I've shown the specimen to lots of like bird paleontologists, and they see it and like right away, like, oh, that's like a foot-propelled diving bird, it's like a loon or something like that. And then they see how small it is. So this bone, it's just like the bottom half of it, but it is like maybe a centimeter in length. So super, super tiny. So it couldn't really be like a foot-propelled diving bird because it's just too small. So we're still trying to figure out what exactly this thing is. So this is some research done by another undergrad, Hamilton, as well as Joe's work on it too. So we CT scan, micro CT scanned it up at Princeton 2 years ago now in the Griffin lab. So it's a photo of the specimen and that's our CT scan. And then we can also look at kind of the cross section of it. So that's what you see in the top there. The black is bone and the gray and white is just empty space. So it's fairly thick for the typical bird. So that suggests it's more in line with birds that are spending time in the water. So it does seem to be like an aquatic species. So now we have to kind of run some phylogenetic analyses and see like what species it's most closely related to. And hopefully that'll help us figure out what exactly this thing is. So like, best guess at the moment is probably some little shore bird that's spending time on the beach or maybe like mangrove or lagoon area.
Are there a lot of these bones that you see this size, or it could be just one little chick?
Yes,
Just
so
size-wise.
from what's preserved, all that amounts to this element, so these little bits down here are fully fused and the bone is smooth, so it suggests that it is an adult individual. And also most bird species reach like adult size within like the first year or so. So it seems to be an adult, so it's not just like a little baby. So this is the only bone we have. There's a few other bird bones at Sea Academy and in Jason. Those are all much bigger and different morphologies, so it's definitely like not anything new or not anything that's already known. So it might be a new species. It's just like I don't know if we have enough to like name it. We could, but usually kind of frowned upon. Yes.
Have you checked the modern bird collection here yet?
No.
Delaware, I'm a volunteer here. Edelmen has one of the largest bird collections in the country.
Okay.
So if you're trying to figure out what kind of a bird it is from modern specimens, I would talk to the curator of birds here.
Okay. Definitely much out there. Yeah. Thank you.
Years ago, when inverse hand was running, we had a reject pile.
Yeah.
And you hunted the reject pile, you find little bird bones out of, whatever layer they were getting the green sand out of, you would find little bird bones and that stuff. And again, they were tiny, tiny bird bones.
Yeah, the stuff at the New Jersey State Museum, typically bigger. They have like some wing elements. There's two other tarsometatarsii. One's pretty big, and then the other one's small, but it's still like much larger than ours, and it looks a bit different. So it's not anything that's already known. So if we can, it'd be awesome to name this new species. It's just if there's enough diagnostic features present to do so or not. And then aside here, some future projects that will hopefully get done this semester. So if you've been to a museum, you've probably seen our two three-dimensionally preserved fish. So one is there on the top left. So it's a bit hard to see, but you're looking at the right side of the fish. So the skull would be kind of on this side, and that's like the gill arch region there, and then the body is off to your left. So if you flip it over, you get scales in place on the other sides. So you have that one, and then another larger specimen. Again, we have partial skull preserved, and then part of the body preserved in 3 dimensions as well. So a couple of years ago, some other researchers named a new species of fish, Eridopristis harrisi, from a few 3D fish at NJSM. So we have 2 in our collection. The researcher who named that new species kind of thinks that this particular specimen might also be Eridopristis, but our larger one might be something different. So really cool for another new species of fish. So yeah, we're gonna CT scan our 2 fish and just gonna see what those 2 species are. Hopefully one of them is new to science. At the very least, it'll give us a few new bones for our known species of fish. And then future research project beyond that is just kind of figuring out like why are these fish being preserved so exceptionally well. So we have these 2. These are actually found over by kind of the drainage pond pump house, very close to one another. So we have those 2, and then a few at NJSN. So it's like, is this particular species being like preserved in 3 dimensions, whereas like every other fish we find is like, you know, a tooth or a few scales. So another interesting taxonomic question for that one. Next one up here. So this very large mosasaur tooth that John Wolfe actually discovered years ago when he was digging with us. So that's actually our largest mosasaur tooth. But when we kind of were putting it together, we saw these little tiny like borings throughout kind of the roots and then up through the crown of the tooth. And we see some weird damage on other teeth and bones. So we want to CT scan that one and kind of just see what the shape of these borings are. And then I'm hoping or hypothesizing that this might be evidence of ostodacts, so bone-eating worms. They're already known in the Cretaceous from some other sites around the U.S. and a few parts of Europe. So probably expected to find them here. We just don't have the evidence for it yet. So yeah, looking at those, we have lots of wood and a handful of seeds and bits of amber that need to be identified as well. So that's good size cedar fruit that was found at the fossil park as float, but we have a few from our fossil there and tons of wood. So identifying those, so our next step, I assume we're working on this semester.
What's the scale on that?
So it's centimeters, so it's about maybe 1 inch and a half, 2 inches. And no one has really done much work on fossil plant material from like South Jersey. Most of the research was done the amber from the Sayreville sites, which is really productive. They found lots of like insects, like a feather and some other really cool stuff in that material. But not much has been done for like cornerstone plant material.
One of our members found a piece with 9 feathers in it.
Oh, nice.
It's at the American Museum.
And then another one, copper light down here, so that's poop. That one's from a shark spiral that went in, so probably came from shark or cartilaginous fish at least. But we have a few of them that have bits of fish bone kind of embedded within them, so it's whatever fish or animal that shark ate and pooped out. So we want to CT scan some of those coprolites to see what else is preserved in them, and if we can identify those little fish bits, then we can kind of start to reconstruct the food chains there. So at the very least, saying like this was a free species for sharks. Yeah.
Maybe I misunderstood the ecosystem. Is this aquatic, or you said there's a lot of tree woods?
Oh yeah. So sorry. Yeah, it's basically the bottom of the ocean. Coastline probably somewhere near Philadelphia, so we're a couple miles out to sea.
Okay.
So all the plant material, the wood and things, were just things that washed out to the sea.
Driftwood.
Yeah.
Floating seeds. Okay.
Yes. Yeah. So things probably growing in like Philadelphia, Pennsylvania, Delaware, that
Okay.
during a storm or whatever just got washed out. It seems like you, like, in like major rainstorm, you'll see like trees floating down like from rivers through Philly.
But they won't let you dig up Philly,
Yeah.
so, got it.
So yeah. Yeah, and the same with some of the terrestrial animals, like those freshwater turtles, and died on land, got washed out in the sea, and then like the Hadrosaurus and Dryptosaurus that are found in South Jersey. Again, living on lands, they died, snagged by a river or something, and just got washed out
Okay.
to the sea. Yeah, so we have lots of interesting projects. I think I have like 4 or 5 undergrads doing research with me this semester, looking at some of these different things. So it's a really active research program at Rowan and Edelman Fossil Park. So that's all I have for today. So again, thank you for being here. Thank you for having me.
I'm not, this is not my technical space, but there is some information that the northeast of the U.S., the Middle Atlantic, was part of Africa, and then it drifted apart. Are any of these remains from, or was, when the continents drifted apart, there wasn't any plant or animal material?
Yeah, so Pangea, that's like Permian, starting around the Permian. So we do find like some of the same species in like South America and Africa, so like plants and animals. But by this time, like the continents are pretty close to how they are today.
Okay.
So we have like aquatic things that could like swim across the ocean. So we do find like kind of the same or similar species in general, like at our sites as well as like other parts of the world. But
And the Ice Age was much later, so
yeah, so
these
just
fossils are much older.
yeah, so our fossils are about 60 to 66 million years old, and then a lot of the Ice Age fossils you find, it's mainly off of like the coast of New Jersey. Those are probably more like 10,000
10 to 20?
years. Yeah.
You may have said this and I didn't get it. Your turtle taphonomy?
Yes.
That
Yes.
was the comparison of the structure of the thin sections. Is that taphonomy the word for that?
So that
Well,
would be histology, yeah,
histology.
histology for the bones, yeah.
Have they done that for modern turtles?
Yeah, so that's what Shire's initial research
That's what
was.
that-
With,
oh,
yeah, so
okay.
it's based off like thin sections some modern turtles and like knowing where they actually live, and then applying that data to the fossil record.
So by looking at the modern
Yeah.
trends, you could maybe match with
Yeah.
your fossils with similar.
Mm-hmm.
Okay, all right, thank you.
Yeah.
Do you have a website, and do you have the name of the person doing the plant research? I like plants, I like ancient plants, so I would
Yeah, so
consider reading it.
it's actually one of my undergrad students
Okay.
that's doing that plant research. But
Mhm.
yeah, right now we're just trying to like find any relevant.
Okay, you don't have like a
papers.
website or updates
No,
or anything, okay, or person's
fossil
name?
Yeah, we post some things on the fossil park page, but
Okay.
yeah, it'd be good. Yeah, I do that. Get one of those websites set up. I'm focusing on all the research.
Question. In one of your early trips, you showed Wiltonia. I didn't know that was in North America. It's a common species in North Africa.
Yeah,
Where else does it occur in the U.S.?
I would have to double check. That paper is freely available, so if you just like, Google search it, you can download
Oh,
it for free.
yeah, okay, so it's
Yeah.
got your name on it, so yeah, okay.
Yeah. Yeah, so it goes over all this
I'll
stuff.
take a picture of it so I know what to look for.
Yeah.
It's on the Facebook video now.
It's on the Facebook video.
It's on the Facebook
All right,
video.
I'm good.
Yeah, we also have like updated font lists for the fossil park and the different formations as well.
Neat.
Any other questions?
Yeah, I'll give you one. What's your coolest like turtle facts? Your like ancient turtle facts, or your favorite ancient turtle facts? Anything. Or were you like, yeah, this is super cool?
Yeah, no, yeah, turtles are just like super weird because they have such like unique body plan.
It doesn't have to be your research, it can be anything. I don't know anything about turtles.
So I know that they're the only vertebrate species where their like shoulder blades are like inside of their rib cage. So like ours, you know, are on our back right there, but theirs are actually insides. But yeah,
Oh, that's interesting. I don't even know how
it's
that
hard to
works.
say.
Okay.
Not really
I'll remember that though. Tell somebody else.
It's a cool specimen at the Academy. So it's one of the really large species of turtle. I can't remember what that name's similar to Partholon, that like enormous prehistoric one. So they found is the humerus, partial humerus, 100 or so years ago, and then a couple years ago now, someone else found another half, like a very large humerus. They took it to the Academy, and two bones like actually fit together. So it's, it appears to be like two different parts of the same bone found like 100 plus years apart.
Wow. That was crazy.
Yeah.
And the shoulder was inside.
Again, thank you. Good introduction.